Good afternoon, students. Welcome to the online class of Biology for Engineers. In our last session, we have seen the topics related to Unit 2, Biodiversity. In the Unit 2, we have discussed about the plant systems. We have seen the basic concept of plant growth. Then we have seen about the different types of growth which is seen in plants. Then we have seen the necessary conditions for plant growth: water, temperature, light, oxygen and nutrients. All these are important for plant growth. Nutrition. The process of nutrition is the uptake of nutrients or the elements or minerals for the growth and development, which is taken by the plants from the soil. We have also seen the role of the macro and micronutrients. Nutrients, which is important, and they are performing specific functions, different activities in the plant. So we have in the Unit 2 three systems. Plant system we have discussed in our last class. Animal systems also we have discussed, and the last topic which is remaining in this Unit 2 biodiversity is microbial system. This microbial system is the last topic, and if we do this microbial system, we'll finish off the Unit 2. So this microbial system, it mainly deals with the microorganisms like bacteria, fungi, virus, algae. All these microorganisms will be included in microbial system. It includes the study of the microscopic organisms, the history of microbiology or the history of the microbial system. We'll see here. When we talk about the history, different scientists have observed or done the research on different microorganisms with the help of the microscope. Microscope is the instrument which we are using to do the research on these microorganisms. As they are very small, they cannot be seen with the naked eye. We need to see under the microscope. So history of microbial system. In the year 1546 AD, all these are the important scientific events in microbiology. The research done by different scientists in a particular year is given here. The scientist, physician Girolamo Fracastoro, he has done his research and he has suggested that the invisible organisms are mainly responsible for the cause of disease. As this microbial system is dealing with the microorganisms and they are very small, bacteria, fungi, and they cannot be seen with the naked eye, they are invisible, and all these bacteria, fungi, algae, virus are causing different diseases. They are mainly responsible for infections or the disease. This was said by the scientist Girolamo Fracastoro. Then the discovery of first microscope was done during the year 1590 to 1608 AD. Then Antony van Leeuwenhoek, scientist, he has first observed the microscopic organisms and he has discovered about the animalcules. Here animalcules means bacteria, virus, fungi, all these microorganisms, he named them as animalcules. He used this word animalcules for all the microorganisms. Who has used this word animalcules? Antony van Leeuwenhoek. He has discovered all these small animals and named them as animalcules. The bacteria, fungi, algae, all will come under the microorganisms, and he has used this word animalcules for all the microorganisms in the year 1676 AD. Antony van Leeuwenhoek. He was the first to publish about the animalcules after observing under the microscope after its discovery. Then 1765 to 1776 AD, Spallanzani, scientist Spallanzani, he has disapproved or criticized the theory of spontaneous generation. Spontaneous generation was a belief that the living organisms are mainly arising from the non-living matter. Spontaneous generation theory gave the false statement about the living organism, and Spallanzani, he disproved this spontaneous generation theory in the year 1765 to 1776 AD. Then in the year 1786 AD, Muller has first given the classification of bacteria. Then during 1798 AD, scientist Edward Jenner, he has introduced about the vaccination. Vaccination is the process of giving the vaccine. Vaccine here is nothing but the microorganisms like virus or bacteria. But in the vaccine, we'll use the microorganisms like virus in a weak form or in a killed state. And this vaccine will help the immune system of our body to develop protection from different diseases. So this vaccine will give us protection from different diseases. It will protect our body from different infectious diseases. This was introduced by the scientist Edward Jenner. He has introduced the vaccination process and he has developed the vaccine for the disease cowpox and the smallpox. Smallpox and the cowpox are the infectious diseases, names of the diseases. Then in the 1839 AD, discovery of cell was done. Cell we have discussed in unit 1. Two types of cells we have discussed, prokaryotic cell and the eukaryotic cell. So the discovery of cell was done in 1839 AD. Then talking about the history of microorganisms or the microbial system in the year 1857 AD. Louis Pasteur, he has given the connection between fermentation and microorganism. He has said that microorganisms are doing the process of fermentation. The connection between fermentation and microorganisms was given by Louis Pasteur, scientist. Fermentation here we use in different industries for preparation of food by the use of microorganisms like bacteria, yeast. At industrial level, in the food processing reactions, and for pharmacy in the pharmaceutical industry also, for getting the production of the products related to the pharmacy. Then in 1861 AD, Louis Pasteur, he has disapproved this spontaneous generation theory. Spontaneous generation theory gives the statement as living organisms are arising from non-living matter. For example, people believe that mice, it is arising from the inorganic matter like non-living matter like cloth and with wheat. If both are combined and left over, then it is giving rise to mice, which is a wrong statement given by the spontaneous generation. So this was finally disapproved by Louis Pasteur, scientist. Then in 1867 AD, scientist Lister, he has given his work regarding the antiseptic surgery. Various antiseptics are killing the microorganisms like Dettol and phenol. And this Dettol, phenol antiseptics we are using for the process of sterilization, killing the microorganisms during the surgeries, sterilizing the skin surface by the use of antiseptics like Dettol and phenol. This work was published by the Lister. The scientist Lister has published the information regarding sterilizing the skin surfaces during surgeries by the use of antiseptics like Dettol and phenol. In 1876 AD, Robert Koch, he has said that the bacteria, which bacteria? Bacillus anthracis is the name of the bacteria which is causing the disease anthrax. Anthrax is also a deadly infectious disease. In this disease, flu-like symptoms will be seen like fever, headache, rashes in the human beings also. And this anthrax disease will be caused when the bacteria named Bacillus anthracis will be entering into the host, and this was discovered by Robert Koch in the year 1876 AD. Robert Koch postulates were published. He has given the suggestions. Postulates are nothing but the suggestions. And in the same year, gram staining technique was also developed. Gram staining technique we are using here to give color to the bacteria. We'll give here a dye. We'll use a dye. That dye will give the color to the bacteria, and it is the most widely used procedure we are using for observing the bacteria under the microscope. After the gram staining technique, the bacteria will take the color of the dye and we can easily observe under the microscope. We have different types of microbes. Microorganisms include bacteria, fungi, algae and viruses. All these are the microbes, types of microbes. In the types of microbes, we have first one, bacteria. Bacteria are the microscopic living organisms. They are very small. They are microscopic. They cannot be seen with the naked eye. We need to see under the microscope. The instrument used to observe this bacteria is the microscope. They are usually containing one cell. We have discussed about the bacteria, prokaryotes in the unit 1. One cell organisms, and they are found everywhere. They are both useful and harmful. Harmful, they will cause the infections or the diseases. Useful or beneficial at industrial level, in the process of fermentation. Example, in the production of wine, in the industrial level, and also in the process of decomposition. Degradation process will be done by bacteria. Degradation nothing but the breaking down of the complex materials into simple ones in the soil will be done by the bacteria. Bacteria we can see in different shapes. They will have a variety of shapes, includes spheres, rods, spirals. Although they are in single cell, but they always appear in pairs or in the form of chains, or they appear in groups. A cluster of bacteria we can see. We can observe the bacteria with the help of the gram staining technique. Basing on the gram staining technique, we can see gram positive and gram negative bacteria. Gram positive will be taking the dye and they will appear in purple color, whereas others will be the gram negative bacteria. Then we have virus. Viruses are the most dangerous and they are pathogenic. They will have the ability to cause the disease. In the science of this microbiology, viruses are the deadliest form of microorganisms. They occur in several shapes and they are widely distributed in nature and they are infecting animals, plants. That's why we are calling them as pathogenic. They will have the ability to cause the disease. Pathogenic. And we are taking it from the Latin word poison. And study of viruses, we are calling it as virology. Viruses are obligate intracellular parasites. Means here, viruses are dependent on host. Example of host we can take is the human beings. That's why we are calling them as parasites. They are dependent on other organisms. They doesn't have the machinery of their own to generate energy or synthesize proteins. So they depend on the host cells to carry out all these functions. So after entering into the host only, the virus will get the energy and they will have the protein synthesized inside the host. Outside the host, they will not have the ability to cause the disease. After entering into the host, they will cause the disease because they cannot synthesize their own proteins outside the host, and they are dependent on the host for synthesizing proteins, and then only they will cause the disease. Reproduction is by replication. Replication process is nothing but the multiplication process where one virus will become into millions of virus. They will multiply inside a host, and the genetic material in them, it can be an DNA or a RNA. Full form of DNA is deoxyribose nucleic acid. Full form of RNA is ribose nucleic acids. Progeny virions are formed within the host cells. Virions are nothing but the virus or the causative agents which is getting transferred from one person to other person, causing the infection or the diseases. Virions, virus, causative agent which is getting transferred from one person to other person or from one host to the new host during the diseases are nothing but the virions. Then next type of microorganism we have is algae. Algae are plant-like organisms. Like plants, they will also have the chlorophyll pigment. Chlorophyll pigment, this is the coloring matter which is helping in trapping the sunlight, and this light energy they are using to synthesize their own food by the process of photosynthesis like plants. And they will store the energy in the form of carbohydrates. Carbohydrates are nothing but the sugars we have discussed in our unit 1. Algae can be single cell organisms, or they can also occur in large numbers, that is multicellular. They usually occur in fresh water or salt water. Next we have fungi. Fungi are the eukaryotic spore-bearing organisms. Spore, here are the structures which is helping them in reproduction process, and the fungi will lack the movement or the motility, and they also doesn't have the photosynthetic pigment chlorophyll. Chlorophyll, the coloring pigment which is trapping the sunlight, is absent in fungi. They are also microscopic, but some can be seen with the naked eye. The study of fungi, we call it as mycology. So all these are the types of microorganisms we have in your syllabus. So thank you, students. We have discussed about the microbial system, the history and the types of microbes in our today's session, and we shall continue with this topic in our next session.